When you think of a natural history museum, you think of this, right? Giant skeletons of dinosaurs, mammoths, and perhaps a whale or two. Now don't get me wrong, these are wonderful exhibits, but they do tend to dominate, don't they? Perhaps taking our gaze away from other important fossil groups, such as the invertebrates, like the trilobites, and of course the fossil record of plants, although the National Museum of Natural History has wonderful displays of both. But there is another world of paleontology that often gets overlooked, quite literally, as this is the world of the very small, micropaleontology. In this lecture, I would like to ask, what are microfossils? I want to introduce you to the foraminifera. I'd like to see how these fossils chart global climate change over 120 million years and what they tell us about the death of the dinosaurs, and find out what microfossils tell us about the way that evolution actually works. Now, there's no fixed definition of what a microfossil actually is. Basically, if it's very small and needs a microscope to be seen, then you can call it a microfossil. Most microfossils are less than a millimetre in size, but some are a lot larger. For example, the limestone blocks that make up the pyramids in Egypt are packed full of the shells of Eocene microfossils called nummulites that belong to a large group of microfossils called the foraminifera. Some of these microfossils can reach sizes of 2 inches, that's 5 centimetres. Not bad for a single-cell protist. Given such a broad definition, basically small fossils, microfossils can come from a wide variety of sources. They are also, not surprisingly, the first fossils we find in the geological record, given that first life was probably microbial. At present, the oldest fossils and microfossils come from the Strelley Pool Formation in Australia. The fossils were originally sulfur metabolizing microbes living in between the sand grains of a tidal beach over 3.43 billion years ago in what is today the Pilbara region of Western Australia. In addition to microbes, microfossils also include many important components of the microplankton, which forms the base of almost all aquatic food chains. Microplankton's sensitivity and reaction to Earth events in the wider Earth system are critical in any narrative we're trying to develop about the evolution of our planet. Planktonic microfossils include those with organic walls, like the dinoflagellates, a group of marine protists that move around using a whip-like flagellum. Many dinoflagellates are photosynthetic, but some are tiny predators that feed on other protozoa. As fossils, dinoflagellates are mostly known from those species that have an encystment stage as part of their life cycles. They vary in size from about 15 to 80 microns. A micron is 1/1,000,000th of a metre. It's kind of difficult to imagine the length of a micron, so consider this. A human hair is about 50 to 70 microns in diameter. Many pollen, such as the ragweed pollen, the cause of much summer allergy misery, are often 20 to 50 microns in diameter. Not all microplankton has a test made of organic material, though. Many secrete mineralized skeletons. For example, the coccolithophores are photosynthetic protistan algae that secrete a series of plates or coccoliths composed of calcium carbonate. Coccoliths have been so productive in the past that they have become significant producers of sediments and carbonate rocks, and today can still form enormous blooms that can be seen from space. In this image, you can see a bloom occurring off the coast of Cornwall in southwest England, close to where I went to university at the University of Plymouth. A good example of very high coccolithophore productivity comes from the famous White Cliffs of Dover that are almost 100% coccolith. These vast accumulations were produced in the warm, shallow seas of tropical Europe during the Cretaceous period. These, though, from an aesthetic point of view, are really some of my favourites. The Radiolaria. These protozoa secrete beautiful silica skeletons, often looking more like Christmas tree ornaments than products of microscopic biology. Together with the diatoms, one of the most important components of microplankton today, they are another important sediment producer, covering the deep ocean floor with a fine sedimentary rock called a siliceous ooze. Larger organisms can also contribute to the microfossil assemblage. Spores and pollen from plants are an important component of the paleontologist toolkit, both for environmental analysis and for the correlation and dating of rocks. We find the first plant spores at about 470 million years ago, possibly produced by the first plant colonizers of the land, perhaps something like modern liverworts. These also provide some of the earliest evidence of land plants that were being consumed by herbivores. As we can see here, this specimen from the Middle Devonian of New York State has a hole and surface feeding trace made by a small arthropod. By the time we get to some of the first plant macrofossils, like Cooksonia at 450 million years ago during the Silurian, we start to see an increasing diversity of spores reflecting the spread and diversification of plants across the coastal landscape. But animals are in on the microfossil game too, and perhaps some of the most famous are the conodonts. Initially, the conodont animal was only known from its conodont elements, these teeth-like objects, commonly about 250 to 2,000 microns in size. They're composed of calcium phosphate. They're found in the Cambrian period all the way through to the end Triassic, which is associated with a probable extinction event at about 200 million years ago. Conodonts are diverse and include many species, some of which had quite short geological ranges, making them again useful in biostratigraphy. However, even though the elements were discovered in the mid-1800s, we still don't know what conodonts actually were for a long time. As conodonts were commonly found in assemblages of paired elements, it was assumed that they formed some sort of articulated apparatus. But it would not be until the early 1980s that the soft parts of an eel-like chordate were found, with conodonts arranged as teeth at the feeding end of the animal. Conodonts turned out to be chordates, not a direct ancestor of us, but certainly one of our early cousins. But let's focus in a little here. I'd like to introduce you to a particularly long-lived group of microfossils that have become invaluable in our understanding of the Earth system over time. Let's consider the foraminifera. The foraminifera, or forams for short, are a group of protists that secrete a shell or test, a mobile part of the foram's cytoplasm called the ectoplasm. No, that's not the stuff that was reportedly excreted by Victorian spiritualists, can develop tentacle-like structures, pseudopodia, that reach outside the shell and capture prey like diatoms and other microscopic creatures. Many forams also contain photosymbiotic algae in the planktonic form Globigerines sacculifera. The pseudopodia contain symbiotic photosynthetic dinoflagellates. These dinoflagellates are transported to the distal parts of the pseudopodia in the morning so they can bask in the sunlight and then retract back into the shell in the evening. Some non-planktonic benthic forms with streamlined shapes actively burrow through sediment, processing organic detritus, while others crawl around on the ocean floor using their pseudopodia for locomotion. Forams are found in many diverse environments, from the shallow to the deep ocean, with some even in moist terrestrial environments. They are found in climates that range from the tropics to the poles. This is a diverse group of very successful creatures. The shells of forams are composed of various materials. Some are made of organic material called tectin, while others are agglutinated or glued, composed of grains either randomly or very selectively taken from the environment and stuck on the creature to form its shell. For example, the foram Technotella has a particular fancy for making a test out of sponge spicules that it collects from its local neighbourhood. Other forams that secrete minerals to form a shell, most commonly out of calcium carbonate, but some species also produce silica. But forams are not just an interesting tiny curiosity. They are a powerful tool in paleontology and Earth history, as they are widely distributed by ocean currents, especially the planktonic forms. They can be found in many different sediment types, making them great for correlation between different areas. And, importantly, they have a relatively continuous fossil record in ocean basins since the Jurassic. They also occur in high numbers, sometimes in the tens of thousands for a relatively very small volume of sediment. And very importantly, they evolved rapidly, producing short-ranging forms that define short packages of geological time. This makes them excellent biostratigraphic tools, and they're used extensively by the petroleum industry. Perhaps one of their most significant contributions is in our understanding of long and short-term climate change in the past. Forams are excellent paleothermometers. Their usefulness in this regard comes from the isotopic composition of the calcium carbonate shells that the forams secrete, and in particular the stable isotopes of oxygen that get incorporated into the calcium carbonate. We are especially considering here the ratio between two isotopes, oxygen-16 and the heavier oxygen-18 that has two extra neutrons in its nucleus. And how does this system work? When the foram is secreting its shell, oxygen atoms from the surrounding seawater go into the newly formed solid mineral. Oxygen-18, being heavier and not as energetic, gets incorporated more readily into the crystal structure. The colder the seawater is, the more oxygen-18 gets included, therefore giving us a proxy for the temperature of the seawater at the time of formation of that particular shell. A change in 1 per mil in oxygen-18 levels represents a change in temperature of about 4 to 5 degrees centigrade. Oxygen isotopes data gained from forams have provided us with insights into the climate change in the most recent era of Earth's history, the Cenozoic, that runs from the end of the Cretaceous, 66 million years ago, to the present day. Let's examine a few points on this graph. At around 55 million years ago, an event that causes a negative spike in the oxygen-18 records of forams records a warming known as the Paleocene-Eocene thermal maximum, where global temperatures soar 5 degrees centigrade, that's 9 Fahrenheit. This warming matches the time of migration of many tropical mammals towards the poles. It also explains the presence of palm trees in Greenland and Patagonia, and the mangrove swamps in southern Australia, which at that time was well south at about 65 degrees south. This warming was possibly triggered by carbon dioxide emissions related to volcanism associated with the breakup of the supercontinent of Pangaea, and perhaps the release of methane, another greenhouse gas, from the oceans. As forams are distributed vertically through the water column, they also provide a valuable picture of what parts of the ocean were warming. In this particular warming event, planktonic and benthonic forams show that the entire water column, and not just the surface waters, were experiencing warm conditions. The forams also record a major cooling event at the Eocene-Oligocene boundary, about 34 million years ago, with a sudden positive excursion in the oxygen-18 isotopic record. This signals the development of the first major ice sheet in Antarctica, as the continent drifted to the South Pole and started to become isolated by the Antarctic circumpolar current. There is another drop in temperatures here at about 14 million years ago, called the Middle Miocene climate transition, which by 8 million years ago would see temperatures drop to levels that would establish ice cover present at the current levels on Antarctica. As you can see, the overall trend is one of cooling and increasingly positive oxygen-18 oxygen values, all faithfully recorded by our tiny paleothermometers, the foraminifera. So that is the picture from the Cenozoic, but can we take this back any further? This is just what Dr. Brian Huber of the Department of Paleobiology has been doing, pushing the record of temperature changes back 120 million years, well into the last period of the Mesozoic era, the Cretaceous, when dinosaurs still ruled the land and ammonites swam through the oceans, keeping a wary eye out for the scary giant aquatic reptiles of the time. One of the reasons Dr. Huber can make this trip back in time is due to the nature of the geological materials that he samples. One of the largest depositories of Earth's sediments, forams, and therefore climatic records are the oceans, faithfully recording information layer by layer like the pages of a book. As we know, we live on an active, dynamic planet, and oceans are continually being opened up at mid-ocean ridges, but also destroyed at subduction zones. When the latter happens, the record is destroyed or so altered by heat and pressure that sensitive isotopic information is lost. The oldest piece of active ocean, one that is still generating new crust, comes from the Western Pacific and Northwestern Atlantic, dating to about 180 million years during the Jurassic period. You can see it here on this image, with the young, newly formed ocean crust coloured in red, concentrated around the mid-ocean ridge systems, and the older crust coloured in blues and greens further away, demonstrating how they've been moved from the ridge systems to their current locations. For the Atlantic Ocean, this operates about the speed that your fingernails grow. The older you go back in time, though, the fewer sedimentary sequences in the oceans you're going to find. And when you do find them, there will be a greater chance that, over time, these sediments and the fossils they contain may have been altered, destroying the precious isotopic signals you're looking for. Fortunately, Dr. Huber has found a number of locations where Cretaceous sediments are preserved, and the level of preservation of the forams is excellent. Much of the data he has retrieved for his studies of the Cretaceous has come from the Integrated Ocean Drilling Programme, the IODP, an international marine research programme that specialises in taking samples of sediments from below the seafloor by drilling. Using ships like this, the Resolution, sponsored by JOIDES, the Joint Oceanographic Institutions for Deep Earth Sampling. From the forams he has collected, Dr. Huber has extended the climate record we were looking at for the Cenozoic, starting at 66 million years ago, back another 55 million years to about 120 million years ago. Just look at the oxygen isotope data and those low to negative readings. Remember, the more negative the oxygen-18 signal, the warmer the climate. So if you thought we hit warm times during the Cenozoic, you ain't seen nothing yet. Get ready for the Cretaceous super greenhouse. Here is the world. Around 90 million years ago, sea levels were far higher and there was probably no ice at the poles. As a consequence of those high sea levels, many of the interiors of the continents were flooded. See how much of Europe is reduced to a series of tropical islands, and that much of what is today the Sahara Desert was occupied by a warm, shallow sea. North America at this time was split in two by a body of water running north to south called the Western Interior Seaway. That explains why you can find rocks, originally marine sediments, dating between 87 to 82 million years ago, called the Niobrara chalk, that is found as far apart as Kansas and British Columbia. Marine creatures like this mosasaur could have swum from what is today the Arctic Ocean all the way to the Gulf of Mexico. Dr. Huber and his colleagues have uncovered a remarkable record of climatic changes that you can describe as varying from warm to very warm over 55 million years. Let's focus in on some specific points that illustrate just how useful these microfossils are in paleoceanography and climatology. One of the events that Brian was interested in occurs between two stages in the Cretaceous called the Albian and the Aptian at around 112.5 million years ago. Sediments taken from the ocean off the coast of present-day Florida, called the Blake Plateau, provided a record of sedimentation across this interval. The boundary between the Albian and the Aptian is recorded by a sudden colour change that you can see in this sediment core. At the same level, there is an extinction in forams and a general change in their size across the boundary, from large in the Aptian to small in the Albian, with the diversity of planktonic forms reduced as well. At the same time, there is a negative shift in the oxygen-18 isotope data in the foram test, indicating that the oceans were getting warmer. As well, there is a corresponding shift in the carbon-13 isotopes, which is generally associated with environmental problems in the biosphere, something that we covered in another lecture in this series. That sudden colour change is actually a little worrying. It could mean that a period of erosion had occurred, removing sediment and accounting for the sudden changes in the fossil assemblage and isotopic data across the horizon. Such a gap in the geological record is called an unconformity. In fact, when analysed, it was found that there was at least a million-year gap in deposition, which might account for the sudden changes we were recording there. Fortunately, there were other sites to study. For example, on the Falkland Plateau down in the Antarctic, where a complete section, without an unconformity, confirmed the extinction event in the foram fossils and their isotopic shifts. Given that the same signal was also found in the southern hemisphere, we can determine that we have something of a global event here, something impacting the entire biosphere, forcing microplankton into extinction and warming the oceans. As of yet, we don't know what caused this event, but it does show how the study of forams is steadily adding more detail to our story of our planet. Another extinction was recorded around 94 million years ago, when a number of well-established forams disappear. The deeper ocean benthics formed off the coast of Florida record an increase in temperature of ocean bottom waters of up to 19 degrees centigrade. That is very warm indeed. Closely associated with these extinctions is evidence of substantial volcanic activity related to the formation of the Caribbean Plate, a large igneous platform and a major outpouring of basalt, thought to have erupted around 4 times 10 to the 6 cubic kilometres of material, and likely as the source of the warming through carbon dioxide emissions and the cause of the extinctions. The forams provide more detail about this extinction though. In addition to recording warming temperatures and extinction of species, the pattern of this extinction has a tale to tell too. It would appear that the forams that suffered the most were deep-dwelling planktonic forms. Foram species are often distributed based on bathymetry, that's water depths, with certain species occupying certain depths in the water column. The loss of an entire group of species associated with a particular level in the ocean suggests that there is no longer a sufficient ocean temperature gradient to support as many diverse depth-related species associations. That is, the ocean is becoming more homogeneous in temperature, leaving no room for these particular deep-dwelling, warm-sensitive forams. This demonstrates the power of these amazing little fossils, for not only are they recording shifts in temperature and the timing of the extinction, but also the detailed ecological factors involved in their extinction too. But thinking about extinction events, how do forams speak to one of the most famous, the one that killed off the dinosaurs? The end Cretaceous extinction event is probably one of the most well-known crises in Earth history, most likely because of its link with the death of the dinosaurs and the massive impact centred on the present-day village of Chicxulub on the Yucatan Peninsula 66 million years ago. The story of this extinction is far from being completely told though, as forams were and still are an extremely important part of the ocean system. They are useful during events like this, both for timing and providing insight into the causes of the extinction. One of the questions regarding the Cretaceous-Paleogene extinction concerns the state of the biosphere prior to the Cretaceous-Paleogene boundary. The vast majority of scientists accept that an impact occurred at the end of the Cretaceous and that it had a severe and detrimental effect on the biosphere. But what was the cause? Was that the only cause? An additional finger of blame is often pointed towards India and a sequence of rocks found on the Deccan Plateau called the Deccan Traps. Trappa means step in Swedish. They are vast outpourings of basalt lava that occurred at the end of the Cretaceous, thought to last around 30,000 years, producing lava flows that might have originally covered around 1.5 million square kilometres. It's not lava that's a problem though. Creatures can always migrate away from centres of volcanism. It is the carbon dioxide and associated global warming that could potentially cause the most serious impact to the biosphere. So what do the forams tell us? Dr. Huber's research shows that warming started around about 65.9 million years ago. This trend began just before the impact occurred at Chicxulub, with a definite shift in oxygen-18 values towards the negative. This temperature increase reversed a long, slow cooling trend that had been progressing throughout the late Cretaceous. The forams don't record any major extinction throughout this interval of time, although sites close to the Antarctic do record temperatures rising, perhaps as much as 15 degrees centigrade. Species that previously existed at low latitudes had started to migrate to higher latitudes as ocean temperatures warmed. But there does not appear to be any major global stress in the foram populations at the time. This warming is also registered on land too. For example, North Dakota, which today has an average temperature of around 43 to 45 degrees Fahrenheit, had an average temperature of around 65 to 68. Even more impressive is when you realise that North Dakota at this time was about the same latitude as Quebec City is today. Imagine Canada with alligators and palm trees. Overall, although there were definitely shifts in the distribution of species, it does not appear that the biosphere was particularly severely impacted during this warming. But what about the meteor impact at Chicxulub? Again, the forams tell an interesting story. Cores taken from the Pacific and Atlantic Ocean will often tell a familiar story across the Cretaceous-Paleogene boundary. A dramatic change in strata from white chalky sediments into a much darker horizon that contains molten material that was sprayed out of the impact site, some of which fell into the oceans. These strata are capped by a thin, rusty fireball layer representing fine debris and soot that rained down out of the atmosphere after the main event. As we noted, the forams appear to be doing fine before the impact occurred, but after it, they register a 90% extinction of the group, particularly in the planktonic forms. Before the impact, there is a diverse fauna of various types and sizes of foram, and after the event, in the earliest Paleogene, only a few small, less ornate forms survive. For the forams, it was likely darkness that would be the killer. You can imagine fine ash and soot thrown high into the atmosphere would cut off the sun and shut down photosynthesis, both in the oceans and on the land. Like a house of cards, once you take away the food web support at the bottom of the pile, the whole structure, in this case the Mesozoic biosphere, came tumbling down. So microfossils hold great potential in detailing changes in climate and recording the progression of major events in Earth history like mass extinction events. This in part is due to their sheer abundance when compared to large macrofossils. This abundance also allows us to investigate some of the fundamental processes of evolution, an opportunity that was taken up by Dr. Jean Hunt of the Department of Paleobiology. Jean studies ostracods, tiny crustaceans around about 1 millimetre in size that secrete a bivalved organic or calcareous shell. It makes them look a bit like tiny pistachios and are often found from the Ordovician period right through to the present day. When Darwin first proposed his theory of evolution, it was criticised by some as not being supported by evidence from the fossil record. Since Darwin's time, the number of fossils in the collections of museums, universities and research institutes has expanded considerably, leaving no doubt that evolution has occurred, but with the question of how evolution occurs still up for debate. It is this question that Jean Hunt has been trying to answer using ostracods. Does evolution occur in a gradual, linear manner, often called phyletic gradualism, or does evolution progress in a series of rapid pulses separated by periods of apparent stasis with little change, a hypothesis proposed by Niels Eldredge and Stephen Jay Gould in 1972 that they called punctuated equilibrium. There is another process, though, called random walks. This describes how trends in various features of a fossil group can develop that are not necessarily driven by natural selection. In random walks, a particular characteristic of a feature, for example its size or complexity, may increase or decrease randomly if there is no selective pressure. As an analogy, consider flipping a coin. You can get runs of heads or runs of tails, even though over a long time period you would tend to get an average of 50-50 heads and tails. To add just one more layer of complexity to this, though, we must realise that random walks are very rarely completely random. There will often be a bounding wall which will prevent a certain feature varying above or below a particular value. For example, consider the size of ostracods. Although very small, there will be a size below which it will be impossible for these little crustaceans to exist, bounded by such things as functional size of organs or the ability to efficiently respire. This concept has often been illustrated with an analogy called the drunkard's walk. Imagine a paleontologist emerging from a bar, a little worse for wear from too much celebrating after their paper just got published in a prestigious scientific journal. They emerge from the bar and start to walk along the sidewalk, but with one side of the sidewalk bound by a wall. They will randomly wobble along the pathway, but any increase will be away from the bounding surface until the paleontologist in question either gets home or winds up in a ditch somewhere. Gene Hunt tested 251 datasets of the morphological characteristics from 53 different evolutionary lineages of fossils, including ostracods, and found that directional trends only fit about 5% of examples. About 50% could be described as random walks, and 45% stasis with no appreciable trend. This fits nicely with what you would expect from punctuated equilibrium, with most fossils demonstrating either stasis or random walks between punctuated bursts of rapid evolution, with just a minor component of what could be described as phyletic gradualism. Hopefully, these examples demonstrate how micropaleontology provides high-resolution data for our continuing story of the evolution of Earth and the biosphere. Sure, the big stuff is cool, but very often the pages of the story they appear on are written on the pages of the mighty microfossils.